we are going to talk about non-dysjunction. That is how most trisomies occur in humans. And trisomies, you know that with the names like Down syndrome, that is trisomy 21. Other trisomies like trisomy 18, that is Edward syndrome, trisomy 13, Patau syndrome. There are variety of trisomies, and I know there can be patients look in this and parents of patients look in this, and I would like to do this video simply as possible to you to understand. Okay, normal human somatic cells, we name them as 2n. That is to mention, to explain that we have one n from mother and one n from father. N means the number of the chromosome. Usually everybody have 23 chromosome from father, 23 chromosome from mother. So altogether we have 46 chromosome in all the somatic cells. Somatic cells means except the germ cell, other cells contain 46 chromosome. Germ cells is the ova and the sperm cells. Except those, other cells contain 46 chromosome, that is 2n. So what happen in trisomy is they have 2n plus 1, mean they not have they have extra chromosome that can be either coming from mother or either coming from father. So although in previously trisomy 21 was called Down syndrome, it's not something they are lacking. It's something they are having extra one chromosome in the 21 position. Why I call this 21 position? Because we have 23 chromosome in the Down syndrome. Problem is having one extra 21 chromosome. 21 chromosome means there are 23. There are altogether 46 chromosome. 123 from mother and 123 from father. The 21 chromosome is extra amount in Down syndrome. Okay, this can happen with variety of ways. And if we explain again, a normal person have 2n cell, 23 from mother and 1n, that is another 23 from father. So altogether they have 46. In Down syndrome or other trisomies, one chromosome can be more from mother, so 24 from mother and 23 from father, or else 23 from mother and 24 from father. So altogether they have 47 chromosome in the trisomy. Okay, and we try to discuss how this happened. Okay, and how one germ cell got that 24. That is the discussion from here on. For that, we need to understand either in the father cell or in the mother cell, for them to get a 24 chromosome, there can be some abnormality in the division of the cell. Mostly it occur by chance, so it is usually not get inherited with the second child usually, but there's a possibility. That's why we are doing genetic testing. And first we see how this happen in normal cell. Normal cells start with the 2n state, that is they have 46 chromosome, and then they divide into 2 daughter cells, and again it divide into further 2, that is altogether 4 daughter cells. So 2n cell divide into 2. So I put 2 colors because that parent also have that that cell also have chromosomes origin from their father and their mother. So that's why I put 2 colors. Okay, and with the division, the first step is the meiosis 1. I am not going to detail of the meiosis and the meiosis steps here. In the germ cells, it has to undergo meiosis 1 and 2. So abnormalities can arise in the meiosis 1, or it can be arise in the abnormality in the meiosis 2. If it abnormality occur in the meiosis 1, if the abnormality occur in the meiosis 1, that lead to abnormal germ cell formation. Here the normal division, it will divide in the middle in the meiosis 1 like this, and meiosis 2 sister chromatid gets separated. I am sorry, the lower part of the video is missing here. So equal number of the chromatid go to 4 cells, so germ cell will contain n, that is 23. I only draw 3 because of the small amount of area to draw. So we will now see how it goes abnormal. If the abnormality occurs in the meiosis 1, that is the first stage, instead of going like this, the division is going to contain like this. That means instead of 23 and 23 going to separate cells, 23 plus 1 go to 1, and other cell will get only 22. Okay, so meiosis 1 is abnormal here. When the meiosis 1 is abnormal, the person will get 2 abnormal germ cell, because now in the division it's going in the middle like this in the meiosis 2. So 2 germ cells will get n plus 1 and another 2 germ cells will get n minus 1. So if the meiosis 1 is the step that goes abnormal, they get 2 germ cells with n plus 1 and 2 germ cells with n minus 1. Here again, sorry, the lower part of the video is missing, but I know you can understand. It is just a line going in the middle of this cell. Okay, if we take this as the 21st chromosome, he also has the 21 chromosome, he also has 21 chromosome, you can understand first germ cell have 221 chromatic and second germ cell also have 221 chromatic. So then we have n plus 1 germ cell in the first one and n plus 1 germ cell again, n minus. Here the first two has extra 21, second two don't have any 21. So n plus 1, n plus 1, n minus 1, n minus 1. So as we discussed earlier, n plus 1 germ cell with a normal n germ cell, they will get 2n plus 1. That's a trisomy. That's the first way. When the meiosis 1 is abnormal, they get 2 abnormal germ cells with n plus 1. Now we will see how this occur if the meiosis 2 is abnormal. In meiosis 2 is abnormal, they only get 1 abnormal germ cell. So I'll draw it again with putting a green color for the 21 for you to understand it better. Now meiosis 1 is occurring as usual. So they will get identical daughter cells. So I am drawing this here. Okay. So it will be equal. Now meiosis 2 is going to abnormal here. Instead of going in the middle, it will divide the pink color chromatid separated to 2 cells, but instead of dividing here green color one, here it one germ cell get the whole both chromatid there. Okay, so how 4 germ cells appear? I guess first one will get n plus 1, yes, and second one n minus 1, and third and the fourth one will be normal. Okay, so here again we get a n plus 1 germ cell. So with the n plus 1 germ cell joining with the n germ cell, then the individual will get 2n plus 1. That is a trisomy. Okay, you understand, right? Another thing for the biology student to remember, if this is first one n plus 1, n plus 1, 2 abnormal n plus 1 occur when the abnormality occur in the meiosis 1, and when the abnormality occur in the meiosis 2, only 1 n plus 1 germ cell occur. And so it's something to remember, it's easy to remember. If the abnormality occur in the meiosis 1, we get 2 abnormal germ cells with the n plus 1. If occur in the meiosis 2, it's only 1 abnormal n plus 1 germ cell. So this is mainly occur with the by chance, and the chance of occurring this is increased with the parent's age. Okay, but there is another possibility of having Down syndrome without, not by the chance, it's by another process. We'll try to discuss it. And the possibility of having the Down syndrome or trisomy, some people are checking in when the babies are inside the womb, and some people do illegal termination that is not ethical to harm anyone's life. Every person's life is very valuable, and it's not ethical to terminate people, human, just because they are having trisomies. Okay, and most of the time it's occur by the chance, and there's some possibility of having another child like that, but it's not inherited usually. But there's some varieties which get inherited by destiny. Their children are having Down syndrome. That is when the with Robertson translocation like thing, when the part of the 21 is attached to another chromosome and and those type of things we can identify with the genetic testing. That's the importance of doing genetic testing. In that case we can predict their next children, the possibility of having syndromes of trisomies. And at the end, my wish is one day one of you or some of you will able to invent some genetic technique to correct these trisomies and correct these abnormalities occurring by chance in the meiosis 1 or meiosis 2, and that will be a huge achievement of mankind. Okay, thank you very much. This is from Clinical Genetics channel. Okay, thank you. Hope you have a nice day. Thank you.